there are so many good proteases which has been studied and documented. And if you see the classification, serine protease, cysteine protease, threonine protease, acid protease, and metalloproteases. These are the few very classical examples of proteases. And when we talk about proteases, we should not forget that these are a very regular enzyme. It is not a new class of enzyme. Okay, but the function, what is required for drug discovery, has not been taken care in a larger way. So because of the techniques, so the new techniques coming up, new proteases, new functions are being looked for. So if you see in serine protease, chymotrypsin is there, trypsin, thrombin, subtilisin, these are the important proteases. Then cysteine, you have papain, and then viral proteases like which are targeted for vaccine development, okay, for drug discovery. Okay, there is a very protein which is for the HIV1 protease, and then you have this you have proteasome and taspase along with the acid proteases like HIV protease. So the vaccine development or drug development, especially for the HIV or similar type of pathogens, similar type of viruses, they are totally dependent on the drug discovery and other channels which are looked through these proteases. So the structure and other aspects of these proteases are already documented. It is not that these are some new proteases, but the functions, different functions, and their action when you are trying for the drug development, that is also quite challenging. So we cannot cover all the proteases in 1 hour, okay, but these are the proteases where you can focus, and there are some more related proteases, subfamilies, okay, where you can try for more leads for the future drug development. Like if some of the proteases have been worked earlier, try to follow that particular paper and try to see what has not been, where is the gap, okay, what is the requirement of that research. For that, I will take another few slides in after this protease coverage. I will be taking that slide, that will be little long, but I will try to finish in early time. So there you will see the difference, that where you need to focus. The majority of the proteases which are worked needs to be done in conjunction with the protease discovery. So you have to see the discovery, the gaps, and also identifying the gaps and then looking for what has not been described, or if it is described, the angles, what angle they are covering, whether they are covering the angle related to the viral proteins or targeting viral proteins or targeting towards drug discovery or targeting for normal metabolic profiles. So you have to see all those angles together. So in the next, you can see the nomenclature. You go through this slide, multitasking attitude. So if you hold on the work, some urgent work may be unfinished. So meanwhile, you have seen this slide. If you see here, the major thing is that about the reference point, and also why these proteases comes into discovery. In the next slide, probably there is the clue for it. The S1 pocket in different among this chymotrypsin and trypsin, thrombin protease. So these are two important proteases which are always talk in the drug discovery process. So if you see, the major challenge is that about the specific difference between these proteases, and to tackle it, you have to think about what therapeutic need you are looking for. So we took this blood coagulation cascade. So if you see the control, why this coagulation and all those things happening, those who are working more on therapeutics. So if you see this pathway, you can see that where is the need of discovery. So if you see here, this is the factor which is important, and here you have this thrombin and prothrombin which is cross-linked together, and this is a common pathway, and there are many specific factors which are taking care of this complete pathway. So if there is any break, or if you want to have anything related to this coagulation related issues, especially when you talk about therapeutic needs of your research, okay, so you have to think about specific sites where you need to have. Now if you see, there is a damaged surface, and I am not telling all the cascade here, but it is not that well established that this cascade will go ahead similarly with all. This is universal one. Okay. In diseased condition, in a compromised condition, in some conditions where the immunity or immunogenicity of the host is compromised, there may be some changes. That has to be taken care during drug discovery, and that is the factor which is very important when you work on this type of proteases. If you go further, this is another one slide which is telling about the amplification of the response. So you know that why coagulation factor is important, why coagulation is important, and when you talk about the amplification factor or amplification of the response, you need to see that the positive and the negative response, and how this is taken care by the various blood coagulators, and what are being discovered based on those type of research. So if you see here, you have this extrinsic pathway, and there you are trying to see that thrombin and prothrombin together, thrombin and fibrin both together, finding this amplification together, and when amplification is finalized, you are getting the complete coagulation. So the coagulation factor, coagulation of the blood, and also why this is important and why these coagulators are very costly, and doing the drug design in these blood coagulation has major challenge. So more details you can see when you read this paper about that. And this is about the thrombin inhibitors as drugs which are used. I am telling few examples, please note it down. That is dabigatran, then anticoagulant which is provided to prevent the stroke. Okay, you know there are a lot of stroke cases, so this is a very common. Then the oral drug which is in market, that is the prodrug, and it is approved in US in 2010, developed by this Boehringer, where Professor Tong also worked in the earlier days, and this drug is also available in the market since long. So you also see that what is the possibility of reducing the cost of the drug or getting alternate drug. So if you see this drug which is used for the thrombin inhibitors, and especially in the stroke cases. Okay, so stroke cases you might have heard about it or might not have heard about it, but this is very common after maybe 60, and these days even after 55, 60 these cases are happening. So stroke can happen anytime to anybody if they are not having a proper control of their metabolic machinery. And during a stroke, the major challenge is to save the patient, and people they think that okay, these are the blood coagulations and these inhibitors are there, we can try these inhibitors. But sometimes if the stroke is very severe, okay, the whole area get damaged. Okay, so you have to take care of that, whether there are drugs available, and I don't have recent survey of the drugs, but as a tutorial part you can do that. What are the stroke medicines available, quick medicine to just manage the stroke. I am not telling that clearing all those clotting regions. Okay, I am not telling about that, because the clotting of the blood is very high in the regions, many regions in the brain. Okay, so how to avoid that thing? There are so many medicines available, and people they are trying. And if you discuss with any doctor who is taking care of the patient, especially the neurologist, okay, they can tell you the challenges. Okay, and the patients after stroke, they are very slow response, and after 2 months, 3 months also their nature, complete nature is also not like that what happened to be before stroke. So that is another challenge. Okay, so that we are not discussing here, we are discussing about drug discovery. So think about new drug development in this area, that is a very promising area. So the drug available is this one, and this is the human thrombin in complex with this drug, how this drug is effective. Okay, this is about the mechanism, and this paper was published in Nature Communications. And you can see this paper, this is PLOS pathogen paper, where this SARS-CoV-2 polyprotein processing was reported. I am not going on detail because this is not within my preview, so probably it will be covered soon, and I will also be giving you the paper copy of it so that you can read it further. And here the main concern is that these type of protein cleavage sites, as a drug designer who is targeting learning the drug development, in the next slide I will be showing you that how the main protease cleaving site to be selected to have a better drug, so that you have a drug efficacy and drug potency. So these are the cleavage site you can see, and these are the cleavage site which are targeted and have been successfully modeled, and the paper was published by Chen et al. So I am not going here detail. And now coming to HIV protease, this is also a very important enzyme. HIV protease, there are a lot of work is going on, okay. Still we don't have any very good medicine for HIV, okay. So there are a lot of work is going on, and HIV protease, the best part is that most of its structure has been reduced properly and you have most of data available. So whatever you want to do, you want to try, like we did some modeling with HIV protease, VRIP protein, with some docking experiments. So you can do that and try to see the binding pockets, okay, which drug is suitable. When you do the drug likability test and drugability potency test in silico, you can try for this, and drug likeliness properties. Try to see the drug likeliness property of the potent compounds. And there are some natural compounds as well, like saponins, okay. HIV1 protease, very good, saponins. They are found in Solanum tuberosum, okay. And we did some initial work which was published in 2011 or 12 in Nature Proceedings, our lab work, and it was done by not a PhD or postdoc student, it was published by MSc student, okay. So I will share that paper. It was a prelude report, and we could not consider further the work because of certain limitations. But this is a good protease to work on, and those who are working with especially the AIDS drug development, HIV drug development, they should target this protease, and many people are targeting so that they can have the, and this is the structure. And you can see this paper, which is a very milestone paper, rational design of potent bioavailable non-peptide cyclic ureas as HIV protease inhibitor. Based on that, lot of discovery has been done, and this is a very good paper published in Science in 1994, and the paper reports lot of this, and these are the designer drugs. You can see this is reported in Science, some of the designer drugs 1994. And what is the criteria for drug development? Because we are talking since last 4 days about the criteria, I speak on this, I have told you about this, but again to list it on merit, you can see that potency, specificity, solubility, oral bioavailability, metabolic stability, low toxicity, efficacy, and marketability. That is also very important. So these are important criteria, and we are talking about potency and solubility and all those aspects, metabolic stability, okay, that we discussed day before study, what is stability, okay, viability, okay. So these are very important criteria, and I think this is all about that. And now, because we have covered this part, I will be covering, don't know where is the mouse, okay, here. Shrabni, can you help me? Where is your mouse? She is not here? No need to bring anybody else. So this, why your mouse control is not working? Not recognize my fingers. Not my fingers, her fingers, not recognized. That's why I use mouse. Mouse is very important. इसको हटाओ यहाँ से. Okay. So now in the coming 1 hour, say, it may take 1 hour, more than 1 hour, I will be telling you few things which you will not listen in any training program or lectures or something, because I have specifically prepared for the researchers working on drug development, drug discovery, research areas related to structural biology. That is the challenge, and many things are not written anywhere in any book. It is given as so these slides are very important for every researcher who is working either in MSc dissertation or PhD project or a postdoc project or a scientist or a teacher like us. So I used to follow that whenever I give my talk and all that. We follow that. You should also follow that, and that will also tell you whatever techniques you have done in your research. Next PPT.
Next PPT?
Yes, it's done. The first slide, yes. So I will be talking about that, the strategies and actions for value-added research process and scientific communications. When you complete the training, this should be the last presentation, but I am giving it right now because tomorrow whole day Professor Tong will be taking, okay? And this is very important. When I told you this drug is important, that drug is important, this structure is important, training of, but value-added research and scientific communication, this is very important, because I am facing challenge every day, you may face challenge every day as a researcher, and those who are not facing challenge, it means they are either lying or they want to hide with the challenge. So this is a very important thing which I am taking care of that, and probably in the coming slide you will read it. So why academic writing is important? Why we write? Why I am telling make notes? Okay, why you are keeping notes? Okay, why it is important? Because science is knowledge based. Whatever you are learning in structural biology is knowledge based. We are giving you the knowledge, and scientific presentations are short-lived. Whatever I presented, you will forget. Always you will forget, but they are persisting only when you write and document it. If you don't make notes, you will forget. So scientific presentations are short-lived and temporary. They cannot sustain longer. Figures and textual presentations are important. If you see a figure, okay, like you came from a movie, you will remember it. Okay, but if you read a movie review in the newspaper, you will forget. Okay, so this is the same like training you are getting here. The presentations, whatever you are getting, the figures, the graphs, the tables, you will remember. The graphical abstract, you will remember. The DBTL cycle, you will always remember, because it's a properly published, whatever I share. So that's why it is important. And when you go for improving the writing skills, okay, you have to improve it. Improving is just trying for it. It is not that after improving you will improve. You will little improve, okay, then little improve. So dedicate time to write. Biggest challenge with all of us, after this program, if I ask you to write 1 page highlights of the program, most of you will not be able to finish. And if you are able to finish, you will miss something if you have not written. So you have to dedicate time for writing. Whatever you are learning in the drug discovery, structural biology, you try to write it properly. Okay, actually write. You can hear from many students, postdocs, teachers, that mainly students, I am writing, sir, I am writing. If any assignment is given, sir, I am writing. Okay, I am writing. And it is the same from day 1 to day 365. Can you believe that one person can write daily and in 365 days he has not 2 pages complete? It is impossible. It means actually not writing. Thinking he is writing, she is writing, not writing at all. Okay, you can hear this story everywhere in the world, not only in India, everywhere in the world. And this is a very important thing. Actually you write. And I remember we used to write the reports for the thesis. and they give us 1 month, 2 months time. We do in 2 days because we actually do that. Other 15 days we used to see, we make notes and we do in the next 2 days. It is actually 2 days time, but we take 1 month time. So the same thing for you. Many students are doing their dissertation. Okay, it may take 4 hours to write the thesis, 6 hours, 10 hours, 20 hours, 30 hours, maybe 5 days, 1 week. But you have to actually write. If you don't start, you can never complete. That is my complete challenge to you. If you say I am writing, and for that no emails, no internet and no phone. The phone, this one. Okay. So if it is urgent, you can drop the message to your mentor, your friends, your family. I am writing for 5 hours. I am writing for 6 hours. Please don't disturb me. No emails. Emails coming, scrolling emails. It is not possible. For me also. If I want to do there and you see emails coming, so I have to reply the emails, some urgent emails. For me I am telling. For you maybe some other type of emails coming. No internet. Messages which are coming. You are in the drug discovery program, messages are coming. So then reward yourself. If you have completed 1 page, how you reward yourself? Reward means you have to clap yourself. Nobody will clap for you. You clap yourself, then you will get a moral boosting for the next day, next week. If you have completed 2 pages of write-up in a 1 week, and don't copy paste. This I will tell you in the later stage. Okay? Write yourself. Don't use AI. Don't use chatbot. I have seen the many researchers. So when we talk about this program, I thought that it is a very important thing that you are talking about discovery. Okay? You are talking about techniques. So don't use those type of platforms. Even for application CV people, they are using AI. Okay? So this is not good. You use it, but try to understand the essence of it. Okay? And reward yourself is very important. I don't know whether you reward yourself or not, but rewarding yourself is good. Then this is a question. Okay? What are the most important things you focus on as a reviewer when you receive a paper? Being a reviewer, being a author, being an editor, I will show you those rails, specific type of real problems. This is coming when you communicate. You have written a paper, you communicate. So what are most important things your reviewer, your examiner thinks about it? And you can see here reviewer 1, your method section. Okay? Gun point. Okay? Means if you write any section wrong, or maybe you have written it not as per the guidelines or not as per the mandate of that particular research, they will definitely question on you. Okay? Reviewers are working, I will tell you that about that. So when you talk about that, the helpful hint will be that the number 1 reason of paper get rejected during review is because of issues with the method section. Most of the time your write-up, your paper, your research, what you have learned, okay, will be rejected because of the methods. Methods are not properly described. Methods are not properly presented. So what researchers they do, you give less time to methods, which is very important. Methods section is very important. And then these are the key important issues which you need to understand, that what are important aspect in the method. You can't write everything in the methods. Okay? Sometimes you write, I make this buffer, that buffer, I use this ligand. These are not required there. That can go in supplementary. So you have to understand that what sections should come in the methods part. Going further, you see this cycle. Why we do research, scientific research, publications, promotion, recognition, salary increase, grant funding, sharing result with the world. These are the 3 reasons why we publish. Okay? Rewarding. Okay? If you publish a good paper, you get a good position, postdoc position. Sometimes you get incentives in BHU like place where research scholars for writing 1 paper they get how much? You know, incentive scheme of the IOE. How many of you know it? PhD students, they know it. Anybody? So in BHU, if a student is publishing in Q1 and Q2 journal, they get 84,000 rupees. Okay? 84,000 rupees as a research incentive. So getting in BHU is difficult, writing a paper most difficult, getting it accepted again difficult, incentive very easy. Okay? So once paper is done, it's easy to get incentive. It is not a problem. So you see, reward yourself. That is your reward, what I am telling. But it is a major challenge. And then funding, when you write a grant as an individual researcher, like your for postdoc, they will say, okay, propose a project. You have your papers to show your strength. Okay, I know this thing. Then if you say, I know this, I know that, and you don't have any evidence, if I say you, I know this, that, you will definitely go and scroll back in Google. Okay, he was telling this is whether he does something or not. And if you will not find anything, you will not believe next day to me. You will say thank you. Okay? In the same way happens with you, with anybody. If you talk big things, don't do here or don't write it, then sharing results with the world. This is very important. Whatever you are doing, your sharing should be there. Otherwise, how Professor Tong knows me, how I know Professor Tong, how you know Professor Tong, this is all because you are sharing your results with him whenever it is published or whatever activities you are doing. So that is important of scientific research. Then coming back to track research papers, organization of the research paper is very important. After getting, maybe you are trained, fully trained in structural biology after 6 months, 1 year, 2 years. Okay, now you have data, all data of your this proteins, crystallization, okay, docking, simulation, modeling, Coot, all data you have. How to structure it? Big challenge. Okay, I can share personal experience that sometimes students come, sir, I have too much data. Okay, so what does it mean? It means you don't know how to prioritize the data. So when you structure, the organization is very important. Stating, actually write a paper, you need 3 things to figure out your message. What message you want to communicate, your audience, what is your target journal, and your article structures. It comes from the target journal because there is a strict guidelines for the journal. This is the structure. And when you target the journal properly, your audience, you cannot do structural biology and publish in a journal which is doing some research related to microbiology. Okay, you have to see the journals which are taking up structural biology insights. You cannot do that. If you are working on a pathogen biology, especially the physiology, you have to target a physiology journal. So that is very important. Most of the time I will show you that. Please don't start writing until you have some good ideas about these 3 things. If you start writing, you will be failing. Okay, so if you have this idea, that's why ideation I told you about TOC. That is very important. Then documentation process steps. Never trust your memory. Okay, don't think that you are very smart and you remember everything. Nobody is like that. Nobody is like that. I challenge it. Okay, even you have a sharpest memory in the world. Okay, you should not trust as a researcher. So construct figures, graphs, tables as soon as data is available. Sometimes people they say I rush my students too much, or I rush myself too much, because I know I will forget. I know whatever I have to do today, if I am not able to do tomorrow, I will forget. So I don't know about you, but I am like that. And never trust your memory means you construct your figures, data, tables as soon as you are having the experimentation. Maybe draw files, but you keep it. Have all references saved in one folder in your laptop PC. Very simple thing I am written here. Okay, most of the time you save it here, there, desktop, pen drive, that computer, this computer, friend's computer, pen drive, mobile, common happening. Okay, and you have to keep in a folder and then do that. Now, step 1, your analysis is complete. Make some figures and tables. Make an outline for your method section as a timeline. Results, same sequence as method. Keep your introduction simple. 3 paragraphs is the best. 3 paragraph is the best. Don't make a lengthy introduction of 5 pages, 6 pages. No journal is having the too much pages for you. Okay, they don't want to listen everything. Okay, you can see the bigger. Again, I will come to the back to the artistic presentation. Okay, so if the presentation is long, having too much slide, you get bored. Okay, in the same way, for paper introduction also, discuss ranking your outcomes. Very important. Discussion, how your outcomes are better, how your outcomes are discussed in a better way. Then a magical method section is very important. Magical method section. If you write a magical method section, means proper results, proper methods documented properly, then this is going to be a very grand success. Those who have published in big journals, you see their method section. You try to see how they summarize all the methods, all the techniques, and then this section of the paper should take the reader on a journey. How was your data collected, and then how it was analyzed. Results, nothing but results. It means a lot. I have seen many papers, many times you discuss methods in results. Results means results. Don't discuss methods. And I have seen some students, they also write conclusion in results. Conclusion is conclusion. So please understand English, okay? Results is results, methods is methods, and conclusion is conclusion. Don't write results, don't write methods in results. Most of the time it is seen. That is the reason there is a repetition. I used to suggest during presentation, but few students they follow, few of them they so think okay it's fine. Okay, so it's fine because if you are not thinking about it. Okay, so going further, for MSc students who are coming up with presentations, that will be very important for you, and for PhD also, those who are going for the presentations in the coming. Then general format, introduction, what question was studied, this is very important. Material method, what to write, I am explaining that, because otherwise again you will be questioning on yourself. In introduction, what problem you studied, what you wanted to write. Material method, how was the problem studied, you will write in the methods and materials. Results, what were the findings, these are the results. Then what do the findings mean is the discussion. So if you understand this process, you will write properly. If you don't understand, you mix up. Okay, so this is a very general slide. First draft. Okay, this is the biggest challenge. First draft, nobody thinks about first draft. Okay, when you ask your colleague, when you ask your mentor, when you ask anybody who is, first draft is very important. And work with, not against your tendencies, work with tendencies, not with against your tendencies. You have to have the first draft ready. First draft may have errors, first draft may have problems, first draft may have grammatical errors, first draft may not be perfect, first draft may be a basic, but where is the first draft? You are taking too much time in making the first draft, which is very dangerous, especially for the field like this. Why I am discussing here, it's a very emerging area. So what happening if your first draft took 6 months time, somebody will publish the same structure, published, reduced, function explained, you are lost. And then you communicate, they will say, okay, thank you, we have similar paper in the pipeline. Okay, thank you very much. And they politely say you thank you. So thank you because you are not having the first draft. Final draft will come after first draft. But first draft is very important. Start with the easiest section. This is suggestion for you. If you think like you in exam, you choose the questions, okay, I will take question number 5. It is very easy for me. In the same way, choose with the easiest section. Whatever easiest you feel, method section, result section, introduction section, whatever section you feel, not reference one, okay? And you start, not acknowledgement also. Okay, that is most easiest because you know that, okay? Build momentum and keep it going. This is also very important. I need not to tell you, every day I am telling to my students, I am telling to myself, momentum is very important. Okay, you have traveled in a low speed trains, okay, passenger trains you have traveled, and you have traveled in Vande Bharat also, okay. So why the trains, they don't reach on time? Normal trains, passenger trains, because they don't have momentum. Okay, one station, another station, third station, fourth. Sometimes no station, then also stand. Okay, so same way, Vande Bharat, they are fixed. Prayagraj, Kanpur, Delhi, finished. So you have to work like bullet train. You have to work like that. You have to have the momentum. If you don't have momentum, you will not be able to write anything properly. Momentum is very important, and keep it going. Means momentum achieved, but started with momentum from Varanasi, Prayagraj, after that no momentum. Never reach Kanpur. Okay? Never. Or you will be delayed. Same thing. If time is short, emphasize the first draft. Again, I am always telling you first draft. So if you have less time, you have to submit your dissertation, thesis, or you have to submit your PhD or any paper. You have 2 months time, 3 months time, report is due. Okay? You need to emphasize on the first draft. Going further, introduction. It provides background on research topic, what introduction contains. One by one I will cover. I will try to be faster. So, research topic contains a literature review. Keep it focused. Literature review is to be focused. Many times this is a very important section, because sometimes we cite a reference which has not been read properly. We are citing a paper which I have not read. I don't have a copy of it. Somebody wrote that section and I also referred that. You should have that section and make it focused. Identify, approach and justify if necessary. Overall goal is to identify significant problem and explaining how addressing the your research will be explaining that research. Then last paragraph of introduction. This is focus on 3 points. Introduction, they will read introduction and go to the last paragraph. Why you have done this research? Okay, nobody has time, I will show you that slide also. Okay, so you have to justify objective, hypothesis and significance of your research. Mirrors the conclusion paragraph at the end of the discussion. This is a mirror image. Okay, but there you conclude, here you start telling why I have written this paper, why this research was conducted. Okay, so these 3 things are very important. There is exercise, you can do it at your home. Okay, when one should write an introduction section of research paper, in the beginning or at the last or any time. You do exercise and tell me after few months which section should be written first. These are exercise options, you can do that. Okay, I have given you the clue, but it's up to you. It is not that what I am telling is final truth. Okay, I am just suggesting. Okay, you can apply your brain, you can try to exclude whatever you want to do, but based on the experience of many other editors, because I have taken some of these slides, some of these suggestions from some of the prominent editors in the world. Okay, and we have been conducting this type of workshops earlier also. So for this program I kept this because it is very important, and this according to Professor Tong, this area is highly emerging. So every day is very important for you, and if you fail in making fulfilling the deadlines, your research will be outdated after some time. So you have to have you have very less time for that. Now you see this one, 10 steps to successful academic writing. Okay, I say it's an academic writing wheel. Okay, it is a wheel. Okay, and you can see here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 also. Okay, complete wheel of fortune. Okay, so if you see, the fortune of research writing, academic writing lies on this: analysis complete, make figures and tables, structure the methods, structure results, structure introduction. Normally 1500 words they have written. You can have 2000 word also, no problem, based on the general guidelines. Then discussion, then writing methods, then writing results. Picture is worth than 1000 words. So I have seen that sometime I will cover up in the next slide. Sometimes you make a picture also and you write also. It is not desirable. Okay, when you make a table, don't make a figure. If you are making a figure, don't make a table. And if you are making figure, table, no need to write. And if you are writing, then don't make figure and tables. So sometimes student do everything, graph भी बना दिया, table भी बना दिया, figure भी बना दिया, लिख भी दिया. See, this is not required, and nobody has time to do all the exercise. They will see that you don't know the writing. Okay, then writing introduction, then discussion I already covered, so I am not abstract. Why? What did you know? How and significance. These are the three components of abstract. Then title, short, sweet, which attract attention. Okay, I have read few long titles, you will forget. Okay, tightening the screw in PSBA 1. You like the title. Okay, then you will read kya type kar rahe ho. Let's read it. Okay, so novel platform technology. Okay, or certain protein inhibits this part. Okay, so these are the important titles, and we do title brainstorming many times. My postdoc is sitting, Dr. Devas, very experienced person. Mandeep is also sitting here. We do brainstorming of the title, 3 title, 4 title, and then we have one day time, 2 day time to think about it. Sometime I ask him, okay, let's propose 3 titles, and then I mail you. I will also go through it, you also go through it. Which one is the best and which gives the correct message? It is not that you always make a catchy title. Okay, I will show you those titles as well. You go here. Okay, magical methods. Okay, very important. I told you these sections, and if you are writing proper method section, record the details when they are fresh in your mind. Give enough details, but do not describe everything. Visualize the procedure or the process. Construct dummy tables as appropriate. And if you see, this is just supplying incomplete information is not desirable. Describing well-known procedure already described, how the protein is estimated, okay, how the spectrophotometry is used. These methods not necessary to, and failing to describe the control. Control is very important. Many of the time we are not able to take the control samples, so that is very important. And if you are not able to describe this, it means that the research is incomplete. And if you really offer my suggestion, this is the easiest section because you know everything about it. Okay, method section, you know everything what you have done, and you can compile it properly. And the transparent and well written method section will be successful for your particular thesis, dissertation, research paper, whatever you do. Okay, so this is a very important thing. Then results, don't write materials and methods here. Okay, I have seen many people, they write material methods here. Okay, results means results. Okay, usually should not justify experiment we conducted. Okay, many times you start justifying why you have done this experiment and justifying the results. Very little, no discussion should be there. If there is little discussion you can have, but normally no discussion. Usually written in the past tense. Okay, this is a very general guideline. Subheaders often useful. You can have subheadings. Data may be presented in text, tables or figures. Present data only in one form. Often the shortest text section of the paper. One form, text, table or figures. Don't make everything. Guidelines for results. Focus on the data related to stated objectives and hypotheses. Many journals they ask hypothesis, okay, but few journals they don't ask, but you have to keep that. Be selective in terms of data presented. Do not repeat table, figure titles. Explain why points from tables, figures which are not obvious. Make sure text, table, figures are consistent. Okay, 1A, 1B, 1, 2, 3. Okay, they are consistent. It is not that the start the method section or result section you write table 1 and again you are writing table 3 and again coming to table 1 again to coming to table 6 like that. Okay, be consistent so that it is properly well organized. Okay, so this is all here. Discussion component, do not introduce new data. Okay, many times it is happening. Okay, you have written everything. Now if you are introducing a new data here, your discussion is of no use. It is not trustworthy. So don't come up with new data in the discussion. Whatever you have explained, it's fine. It is same like you see in courts. Okay, when the judge all proofs together, then decision come. Don't bring any new proof or something during the decision. So this is your important discussion part, why you are bringing a new data here. Why not you explained earlier? So don't bring it. Present the principles, relationship and generalization shown by the results. Identify the exceptions and unsettled point. Please identify what you have not been achieved or you have not achieved it. Okay, there are few, you cannot have complete successful experimentation. Maybe few things you could not achieve. Okay, you have to mention it. Okay, you have to tell the audience, you have to tell the editor. Okay, so that there are no unsettled issues, all are resolved. Identify the, place the results in context of the previous one. Okay, if previously somebody has done on that protein, they have deduced some structure, you have to mention that. Then identify theoretical implications and practical applications, state the conclusion, what is to be drawn, identify the evidence to support each conclusion. Going further, acknowledgement section. Okay, very important section, but most of the time we forget to acknowledge many people, and most of the time we acknowledge many people who have not helped. Okay, so you have to be realistic. Okay, very realistic. I am happy that you are very big hearted, you acknowledge everybody, but the section should be limited to the real acknowledgement. Okay, so identify the external financial assistance. You are getting some grant, DBT grant or any grant, GRF, SRF, you are not acknowledging it. Okay, you forgot, sir, bhul gaye. So this is not acceptable. Okay, you are being funded by your fellowship, you have to acknowledge. Then grant number is very important, so you ask your mentor, you can ask your office for the grant number, BT, whatever is there. Okay, then thanks for technical assistance providing material or cultures, access to special equipments. Normally you go work, forget. Okay, you write on WhatsApp, thank you bhaiya, thank you didi, and then khatam. Okay, so this is not acceptable. If you have used those facilities, please acknowledge the facilities. Okay, that is very important, it is documented. Okay, then make sure names are spelled correctly. Wahi bhaiya didi wala. ठीक है? You write my name Pratyoosh, YUSH. Okay, please see the spelling name. Okay, you write Nivas, NIWASH. Okay, you add H extra. Okay, he will be angry. Don't acknowledge me if you don't know my name. Okay, you just write XYZ or you acknowledge the department. So please remember the names properly. If you don't know, there is no harm in asking, sir, what is your correct name, spelling name? You write it. Okay, so this is a very simple thing, but it happens many times. You know, we don't spell properly the names. Okay, and some names are very complicated. They have a specific spelling. Okay, like if you want to write Mandip, it can be MANDIP also. It can be MANDEEP. Okay, so what is correct? Monica, MONICA. That is also Monica, MONICA. In the same way, several names. Your name very difficult. Okay, can you spell it? तेज बोलो थोड़ा. हाँ बताइए. I am still, since last few years I am trying to spell her name properly and speak properly. I am not able to do that. But spelling I could remember. Okay, so I will write H in the beginning. Okay, so this is very important thing, but please don't forget it. Okay. Exercise, one more exercise. Why should funding agency be acknowledged? Okay, why? Any answer? I already explained you. Why funding agency should be acknowledged? You will say that it is their duty to help everybody. Why to acknowledge? That is one thing. Okay. The second thing is that funding agency also needs how many funders, how many beneficiaries they have, what output they have generated. So that is required by the funding agency also. Okay, so that's why they will ask. I gave you the funds. Do you have published anything or any details which you can share? And they will see your acknowledgement section. They will not see your method section. They will not see your result section. They will see acknowledgement section. Directly they go there. And if there is there is no name of DBT, DST from where you got UGC, they will say, okay, thank you, we will remember. Okay. and next time they will not have the data about your publication, and they will rank you lower. Okay, that this person has not published anything, though you published. It is a very wrong reflection of your hard work. So please remember this, okay? And you do exercise regularly. Reference cited, okay? This is a very important thing, okay? Referencing. List all significant published references. All references in the text must be in the reference section. It should not be mismatched. Okay, many times reference list mein reference hai, and like we showed you so many papers. If you make a write-up, at least you mention the references which we have shown you. So please mention that. Then check every reference against the original publication, content and citation information. This is very important these days, because during the course of research, many papers are retracted, many papers are withdrawn, many papers have shown some factual errors, so journal have taken back. So if you have cited those papers, it is a wrong reflection of your work. Though you have not done anything bad, but if your paper is having a paper which is retracted, you have to cross check it. It is not possible by end alone, first of all. It is possible one by one checking. So 150 references checking will take maximum 150 minutes, or may say 2 minutes, so 300 minutes, or may say just more than that, maybe 900 minutes, 2 days. Invest 2 days, what is problem? But check references carefully. If you miss this thing, it is not a good thing. Follow the journal's format correctly. Need not to tell you, many people they use the reference as per their wish. Okay, I will follow this, I will follow that. Please follow the reference guidelines given by the journal. Otherwise, what will happen, you will face in the future. Okay. Supplementary material. Okay, this is a very important thing, supplementary material. As I told you, you cannot keep every data here. Okay, I have so much data. I want to give everything to journal. No journal is happy. Okay, they want good data, correct data, properly arranged data. Okay, and sometimes people they use the mailbox as a garbage bin. They throw all the data to the mentor. They throw all the data to their friends. Okay, okay, take your data without arranging, without doing all that. Please use supplementary material feature for accessory supporting data, and it is permitted by many journals. Okay, then supplementary data only, not the main data. Don't mix again your main data with supplementary. It will be supplementary data. Okay, then format varies from the journal by journal. Often website posting is there, so it is posted in the website. Anybody can click and read it. These papers, science papers, nature paper, which we have shown you yesterday, or Professor Tong is referring, they have lot of supplementary data. Lot of supplementary data, hundreds of pages. Okay. They cannot mention everything in the 2-page, 3-page papers. They give you 5 pages to write your results, which you have done in 5 years, 6 years, 7 years. How it is possible? That is the key of success, that you can keep this as a supplementary data. And coming back to the paper again, when you submit a paper, the first revision comes. Okay, if you are very lucky. Okay, otherwise if you are not lucky or there is some problem with your paper, rejected. Rejected is also very common. Okay, so first revision, when the first revision comes, allow time to pass before beginning. Then first draft, rough draft, disjoint word maybe incorrect. First draft are often conversation style. Now the goal is structural alteration. So when you try to do the first revision of the draft, okay, before the paper is being referred, so there are 2 steps. Once the review has been done, first draft you sent, final draft you submitted. Before submission also you should have a first revision at your place. So how to tackle that first revision? That is a very important challenge. So when the supervisor is suggesting you this point, that point, ye karo, wo karo, you have to think about it. Okay, and then try to do that, and you try to have the structured alteration. Now you see, how does the reviewers come and improve the article? When you got a decision, whether the reviewer was very critical, or he she gave some more comments for the improvement of paper, try to see that. Okay, or reviewer editor just direct to include this reference or something. Okay, so you have to see whether really it is worth to read the comments. Normally it is worth. Okay, next slide I will be presenting you overall editing concepts. Target area, editing for the content, is it accurate? Does it achieve the purpose? Editing for organization, how well is the message is presented? Then organization of the manuscript. Then editing for the format. The format should be proper, and also the things are not distracting each other. Okay, they are connected together. Voice, active voice, subject of the sentence performs the action. More precise and less wordy, use active voice. Passive voice also you can use, and you can use that also. Active voice is most preferred form of scientific writing. So be in active voice. Talk to your friends in English, they can also tell you. Title and abstract. Okay, the title and abstract summarize your work, should be the last thing you write. I have seen most of the time you start writing your title and abstract in the beginning. Not good. Mention a tentative title, no problem. You should not write abstract and title in the beginning. Otherwise you will be distracted. It should be last. You finalize last. When you have everything ready, then be specific. Key elements should be included in both. Part of the paper most people will see. This is the part of paper most person will see. Title and abstract. आपको भी मैं जो papers भेजा हूँ कल, you might have seen title and abstract. I bet you. Because you don't have much time to read that. You will read maybe in 2 days, 3 days, 4 days, 5 days. But title, abstract, you might have seen it quickly because it's very short. Title, you understand, okay, what is inside there. In the same way happens with movie. You see the title, okay, Suryavanshi. Okay, you start, okay, Chris 3, Chris 4, okay. They are not. That's why the title is also repeated 3, 4, 5 because you know the story going on. Okay, Batman or कौन सा man है, ठीक है? Spider-Man. So the same thing happens with your papers, and there are some bunch papers coming. Some journals they also take story-based papers. Means one, like if you have discovered any protein structure, second, third, fourth, they will take it. But if it is not a story-based, you don't have the key elements there, and you are not having proper title, then it will not be going ahead. Best way to get people interested. Then self-contained summary of the work and significance. It contains everything. Okay. Title. What should be there? Key element describing the content of the study. First portion of the paper seen by everyone. People judge by the title whether to read the abstract. This is truth. You read the title, you go to the abstract. Okay, in the same way. So if you don't have a proper title, you will not go through the abstract. Many times the paper, there are so many papers coming. If the title is not related to you, not understanding to you, you will never be able to see the abstract. So this is very important. Sometimes people, they think that title, what is happening in title? नाम में क्या रखा है? Okay, in research communication, नाम में ही सब कुछ है. Everything is in the title and abstract. Very important. Okay, so please remember it. Writing a title, okay? How to write a title? Biggest challenge, okay? And tells a complete story. It should not be halfway. Okay, complete story of your paper, what you have achieved. It should be there. Specifically describe the study. Uses as few words as possible. Unambiguous. Includes a model system. Begins with an important word. Concise label, not a descriptive statement. Okay, it should be a concise label, not a very descriptive idea. Okay, maybe I have some tips. Okay, avoid unnecessary word, avoid jargons, be specific. Catchy or cute titles are also acceptable. Fat threats a case of too many hormones? Question marks. Okay, so this is a title which published some paper. Okay, maybe they have described about some specific enzymes. Okay, about diabetes and all those things. Be specific. Okay, pneumococcal responses. Specific stress response of Streptococcus pneumoniae to lactoferrin. So you write specific title. Okay, pneumococcal responses you have written in the title, it is unspecific, not properly telling what type of response. Okay, that should be written. Brainstorming a title. I told you few minutes back, write down as many as type. I am practicing this, so you also practice it. You will be not much successful, but okay, because practice makes perfect. So you try to do that. I and Nivas did it many times. Okay, and with Mandeep also 2-3 times I did, and other students I am doing. Okay, they sometimes think that he is not good, but anyway, I have to think about the proper title. For that, brainstorming in the morning. So sometimes you think, no, it's a waste of time. He should decide a title and give. I go hostel, okay, or I go home. Why 3 title, 4 title? Why should I devote my energy? But brainstorming very important, and put them aside, compose and revise. Okay, compile, again see, then sometime in 2 hours you can get a good title. Sometime 3 hours you will sit and you will go, no final title. Next day you came, okay, something came. Then I used to practice it. That's why I'm telling you. I used to call my students also after some time. Okay, I got this clue which we can include in the title. Then I ask his or her opinion. Okay, we can keep it or not. So these are the things which you should also practice. I am also a student. You are also a student. You will be teacher. I am right now teacher. So this is all which you have to learn. Abstract, self-contained summary of your work, significance, and it should be clear. Don't exceed the word limit. Word limit is there, 250 word, 150 word, 100 word, and 350 word. I don't think that any journal is having 350 plus word. Okay, so sometime you make a big abstract, 2 page. Okay, sir will be happy, you will be happy. Very hard work. Okay, abstract summarized, 250 words to 350 word, and some journals they take 200 word also. Okay, based on the journal guidelines. And some journals they don't ask abstract also. Okay, you start your story. Okay, so they don't give you room. Okay, to write, because they have very limited page restrictions. 2 page. You have to tell your story in 2 page, 3 page. So they have very small section like Trends journal. Okay, Trends series journals, self-press journals. When you write a review, mini review, a forum article, they will not provide you. They will say, I think it was 80 words abstract, 80 words abstract, and we did it. If you read our article, one article, 80 word abstract we have written, and now I know that in 80 words also we can write. Okay, so this is all, but mostly the research data they provide 250 words. Okay, it is not that research you can write in 80 words, but in case they want to highlight 2 points, you have to highlight it. Abbreviation, don't forget to define abbreviation the first time they are mentioned. Okay, DNA, common names, no abbreviation. Okay, DNA, ATP, okay, all those, and some protein specific which are very known. Okay, there is no need to write deoxyribonucleic acid, ribonucleic acid. Okay, okay, scientific writing should be concise, precise, reader based, simple, purposeful, accurate, clear. Okay, benefit of writing already explained you. Final product of your research project, share your result with the scientific community. The more you write, the better you will be. Okay, don't think that without writing you will be a good scientist. Okay, you can never be a good scientist if you don't write it properly, you don't communicate to the world. Okay, you do whatever you work you do. Okay, so that is very important. Exercise, some exercise. Prior to submission, try to get your paper read by some renowned researcher who does not have conflict of interest and is good at editing. I don't know, I could not find much, but sometimes my fellow colleagues they help. It is a time taking activity. Who will do? But you can try. Okay, if you have very good understanding with somebody, some good thing, they can read your paper, they can give you the idea. Okay, and don't be afraid that he or she will steal your data. Please don't be afraid about that. Okay, develop the trust system and try to see, and when you read the paper, okay, when he or she reads the paper, they will be giving you some idea. Okay, and if they are giving idea to you, they are not charging anything. You are taking their time. Okay, so think about that, and that is a good thing because they are the experts, so they can provide more outcomes. Then text tables, graphs, I think I already covered. Okay, one thing I have not covered: text, table, or graph. When to make text, when to make table, when to make graph. Text for simple or non-significant changes. Tables or graph for essential repetitive data. Tables for precision when exact results are critical. Graphs when trends and tendencies are more important than exact values. So when you use graph, when you use table, when you use text, written here. Okay. Tables what and when, use a reasonable number of lines or columns. This is again a problem. Sometimes you make a table, very crowded table. Okay. So really it is required or not? You have to see that. Use for essential repetitive data. If it is repeated, okay, don't write Armstrong, Armstrong, Armstrong everywhere. Okay. Or mu, 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 whatever you write. Okay. So pH, if the pH is mentioned, make a table, write all the pH. pH1, pH2, pH3 again. So same way you have to see, give only correct numbers of significant digits, not 0.000008, at least 2 decimals. Okay. Significant. Okay. Stat आप पढ़ेंगे तो पता चलेगा. Comparing columns is easier than comparing rows. Okay. Must be self-explanatory. Material methods, data and methods. This is a website which I will share you, and this gives you a very important section. Then tables, how to prepare. Title should be brief. Okay, one thing I want to tell you, figure title should be descriptive. Table title should be concise. Figure should be descriptive. Legend should be descriptive. And table title should be concise. Don't do vice versa. Okay. Words usually left justified or centered. This is just a suggestion. Numbers usually right. Footnotes for explanation. There should be NA or something. Not observe anything. You can write. Horizontal lines are okay, but vertical lines are not. Take the journal's format. Figures. Visually the most eye-catching part of the presentation. Figures are very important. Determine the figure's purpose, then determine the form. Don't make all the figures in the main text. Okay. Like you observe some micro, you observe some structure. It can go in supplementary as well. Make sure the important points are covered. Colors can be expensive. Color figures from journals, they take the charge. Charge is written here. We can ask Tong also about it. Okay. So 400 to 800 per page. Color figures. Okay. So you can mention them that color figures I will not want in print. Online it will be colors, but in print form it can be black and white. Okay. So they ask to the choice that your mentor do, but you as a researcher also, you are also becoming mentor in some years. So color figures you can skip that. In print it can be black and white because it is costly. Color figure is online, it is colored. Okay, because without color nothing. Okay. Submission process, I am not telling much detail about it, but it's important. All aspects must be done ethically. Your ethics and integrity are priceless assets. Do not plagiarize. This is a very important thing. Self-plagiarization. You published a paper, again you are making some story out of it, don't want to write the section again. You have to write it. You already got award, reward, whatever you want from that paper. Now you are writing afresh, you can mention the method, have the advance approval for the manuscript from all co-authors. Don't do like that. Okay, parachute authors. Okay, that okay, he is my friend, he is my friend, he is my friend, okay, let them all involve. And then sometimes fight. Okay, so I will not include it. So these things you should discuss in the beginning and have the advance approval. Discuss and try to tell everybody that okay, I will write paper with 1, 2, 3. They all 3 know, and divide the duties also, sections also. In the last they will say okay, she is the first author, she will write everything. And when the revision came, one person will say, sir, I don't know, I am afraid. This happens, I am telling you my experiences. Okay, and then the person will tell okay, the first author will do all the revision. It is not possible because you 3 decided to write, and now the decision is there to revise the paper, or if the rejection is there, you blame the third one. Okay, because of her section I got rejection. So these are things which practically needed to understand. Have the advance approval, which is very important. Decide where to submit. It is also a big challenge. You cannot submit all research. You should have a clear plan where you want to submit. Match the focus of your studies with the journal. Okay, failing to select the best fit could slow publication and results of reformatting. This happened with me recently. Though I am teaching you again, we also fail sometime. So we submitted a paper which is having focus. Okay, I think nature water paper, right? So we submitted to that journal, and they are taking that type of work. We read some work, but now they told after some time the journal also changing the focus. It is not the same focus will be there. 6 months each 6 months try to see the aims and objective of the journal. 1 year, 2 year, they change the focus. I will take this area, I will take that area. So if you are not submitting to the right journal, they will return or slow. No reviewers will be fine. Okay? And if you don't have reviewers, paper will not be reviewed, it will be hanging on, your time will be lost, you will be also frustrated, so many things. Then failing to select the best fit could be the slow publication outcome. Improper journal sequences, no review, what will happen? What I told. If you choose an improper journal, no review, immediate return, thank you. You will get a thank you message. That is 100% guarantee. Okay? Thank you every journal is giving. Okay? So feel good or bad, whatever you decide. Reviewed, rejected as inappropriate. This is also very common. If you submit irrelevant study which is not in the mandate of the journal, they will reject. Poor or slow review, which is very common. Then manuscript published but nobody to read it, because this is not a focused journal. Okay, you publish your study, good study, in a journal which is not having those type of study. Like if I am working on biomass valorization and I publish a journal which is publishing biopolymers, okay, specific polymers, chemistry work, okay, nobody to read. Okay, if like so you need a reader to read it. So reader will be getting from the specific the target audience. The target audience should be able to know that, okay, so if even, so these are the consequences happen. Okay, so your hard work will be go in vain because nobody will read it if it even publish. Normally the first step will happen, so no need to go down. First step will happen or second step. Okay, but if you are very lucky or you are having a very bad luck, both things can happen. Okay, first you will say, okay, our article got published, very good, and then nobody will read, nobody will cite, and if your research is not cited, it means so many things about it. Sometimes this is a discussion debate. Citations, I am not discussing here because of the time, but if your research is not read by people, it means it doesn't attract them properly, or you have not been able to convey the proper message properly. Okay, so this is the reason of non-citation, and citation is a very important thing, especially in the academics. The submission package, the citation, like I say star rating for the movies, right? So if your research paper is good, people will go watch. 500 crore, 600 crore, okay? In the same way like you do the YouTube, reels and all that views, okay? In the same way the paper reading, citation, and sometimes people they read also, it is also coming under that, but they cite, that is the better part. The submission package, I am not telling, but these are the available in the journal guidelines. Cover letter, very important. Cover letter, how to make your cover letter for the journal submission, very important. Spend time in writing the letter, opportunity to address the editor and reviewers directly. This is the place where you can directly address the reviewer and editors. Otherwise in the online, how you can do that? Editor will not chat with you, okay, editor will not reply on email. Once you submit the paper, gone. Whatever you want to write good things about your paper, strong points you write in the cover letter. Explain on more personal level why your work is of great importance and merits for publication. Why this is important, you write in the cover letter. Can be addressed to editor-in-chief. Okay, don't mention the name of editor. Okay, you can write to the editor-in-chief. Dear editor-in-chief. Don't write dear Professor Shukla. Okay, dear Professor Tong, dear, means don't mention it by name, because personal attention is important as an editor. He or she is editor. Okay, so please remember, highlight the article's main point. Don't reuse previous cover letters. Okay, which normally people do. Include a statement that manuscript is not currently submitted to any other journal. State that there has been no previous publication of the type. These are the mandatory important things. Editors and reviewers, this is the series. Okay, there are so many people involved in this process. So managing editor, copy editor, editor-in-chief, editor, editorial board, and reviewers. Okay, editors manuscript receipt, they receive the manuscript, okay, and they communicate with the corresponding author. Editor reviews for appropriateness, then they select the reviewers. Okay, once you submit, I'm just telling you about the submission process. Manuscript then sent to reviewers. Okay, sometimes they don't do, and then it is returned. Review process run by the volunteering. Okay, those who are doing reviewing process, our postdocs, they know it is a volunteer work. Nobody will pay for them. Okay, it is a volunteer work. Okay, the paper came, I need to review. Then usually 2 or 3 anonymous reviewers. Normally 2 or 3, but I have read 4 reviewers also, 5 also, 6 also, 7 also, and sometimes 8 or 10 also. It depends on the journal editor, how many opinion they want to take up. Recently I reviewed a paper, 7 reviewers. Okay, so one of them I was there. So I thought, okay, so many reviewers. When the decision comes, you see the reviewer want to name nahi rata usme. Okay, but you can see the comments, 7 reviewers, and the author who is handling the paper, he or she has to address the all 7 reviewers. Okay, I think with you also it happens sometimes. With you also happen. So this is happening. Nivas also faced this, 1 reviewer, 2 reviewer, and then more reviewers. Okay, length of the time depends on the journal. Normally 21 days, but maybe 1 month, maybe 2 months, maybe 3 months, maybe 4 months, 5 months, 6 months, 7 months, 8 months, 1 year also. Depends on the type of reviewers giving points, good points. Okay, so normally it is 21 days. Normally all good journals, they have a very good speed of publication. So they publish within 3 months after revision and all that. Okay, review guidelines. I think unpublished manuscripts is confidential. So if you have any paper for review or feedback or something, please keep it confidential. Don't tell everybody, okay, I saw this manuscript, I am reading it. Okay, keep it confidential, because the researcher's paper treasure is with you. Don't share. After reading, making understanding of the paper, you should delete it always. Okay, until unless it is published, or if it is dealt in the confidence, that is very important. Don't save in a public computer. Okay, don't share with anybody if you have got anything for review or any feedback. Okay, unpublished manuscript is confidential. Do not discuss with authors. Okay, authors se to never discuss. Okay, but with your colleagues also. Okay, this is the confidentiality. Okay, you have to maintain. So review criteria. These are the criteria adopted by most of the journal. Okay, so same is happening with your PhD thesis. Same is happening with your projects also. And same is happening what learning you are doing in today's GIAN course. After 5 days, what is review? We are doing the review, what you learn. So these are the criteria: research question, extent, experimental design. Especially in drug discovery, experimental design is very important. How well established it is, okay, because it will go for the trials also. So your design should be well established. Then conclusions, discussion, and references. I think I am exceeding the time limit, but no problem. Editorial decision guided by the reviews, and there are 3 basic responses: accept, modify, reject. The first decision is not coming first time. I have not heard any author saying my paper accepted in one go. Okay, most of the time it is modify, and many times it is reject also. Okay, so next slide will tell you the story. This one, okay, happy after 1 revision. Okay, this is my paper. Okay, so I got this paper in 2023, okay, December 30th, last day of the year, okay. So I was like this only, okay, and my co-author was also like this, okay. So you feel very good about it if it is accepted. It is not the first acceptance after revision. So the first decision of this manuscript was modified, revised. So after that we got that, okay, I am pleased to inform you that your article is accepted for this, dear Dr. Shukla, okay, and this Dunia Varga, okay. So you feel it after your hard work, okay. And sometime I have that rejection slide also, probably. No, I don't have. I will show you. Now there is a rejection, so many rejection slides also, okay. I think recently also we got it, okay. Paper get accepted, rejected, accepted, rejected, this is cycle, okay. So you should continue. I will tell you the motive behind that. Don't look for the bias or intent. Sometime as an individual we think that, okay, this person is biased, that person, that reviewer has not seen this, he has not gone through my this data, my that data, I did hard work, every day I come in lab. What is the fun in that? Why he or she should be bothered about it? If you have not done all those basic homework, they will reject. Begin with the presumption that reviewers were unbiased. Think about that reviewers are unbiased. They don't know you. Okay, why they will be biased? Okay, and they put substantial unpaid effort to understand your work, and you are starting or assuming that they are biased. Don't think like that. So this is a good thing for a scientist, for a person who is working in any area of biotechnology. Please think about that the review process is unbiased. Okay, reviewers, they are totally unbiased. Think about it, okay. Even if you see any biasness, maybe if you observe, you ignore it, try to see what is good about it, okay, and try to improve. After receiving your reviews, read and get back, put reviews away for 1-2 days. This is a very important sentence. Reading. After you see the comments, try to read the area what you have done, okay. Try to read it properly and don't see the reviews for 1-2 days, because again you will be biased about that review. So read those areas to improve and then put reviews for 1-2 days, reread the reviews again, okay. Once you got that, you read again the reviews which you have kept for 2 days, then try to understand what reviewers are saying, what reviewers are telling you, what message they are conveying. Discuss reviews with collaborators. This is the most tough part. Most of the time we don't discuss, okay. We feel sometime little not comfortable, okay. I am not discussing. So if you don't discuss, you don't get the clues, okay. Then look for the clues. Editors, cover letter, reviewers is always right. Like boss is always right, reviewer is always right, okay. So if they have decided these are the comments you need to address, you have to address. If you don't address, they will not send the journal for acceptance, okay. So they are always right, okay. Response to the acceptance letter. Once your paper is accepted, celebrate. Very common, like we did, okay. Wait for proofs. Acceptance is much more common following the revisions, okay. So you can do that, but wait for the proofs, then celebrate. Get it published, then celebrate. Little wait is fine, okay. Because sometime during proofreading also there may be some changes, some delays, and there are few journals also, they also revert back decision also. You know it or not, I don't know, but it is there. Revert back in the decision. So celebrate after the proof arrived, after that, but anyway, it's a good time, but normally you get the proof after that. Exercise what can be the reason of rejection. Share your experiences with each other. Okay, there are many people who are here. Okay, what are the reasons of rejections? Proofs, a preliminary copy proof should be read carefully. Answer all queries, usually cannot make drastic changes. Don't try to think smart and make drastic changes in the proof. You can't do that. You cannot do, otherwise your paper will be revert back. Only the typos changes, only the normal editorial questions you can do that. No major changes. Answer all queries. There may be lot of queries, sometimes 100 queries in your paper. usually requires peak, often 48 hours. Okay, this time is very important. Proof 48 hours means you have to keep aside other work on the proof. Okay, you can take more time also. If the paper is long and you need some time, you can take time. Okay, but 48 hours is given for the proof. But you read, this is the last opportunity to correct your work. If the spelling of Rikta is not correct, you can correct it. Okay, but if your data is incorrect, you cannot correct it. You have to put appeal, okay, that this data. So this is very important. You have to have only the mistakes which are very common and editorial, mainly editorial. Okay, so before that submission you should check all that what I told you. Accepted with revision. This is rewrite the sections, give manuscript someone else to read. If you decide not to revise the manuscript, contact the editor and withdraw it. Okay, if you want that I don't want to revise, I will submit afresh, you can do that. Okay, but normally this is not recommended because the time of editor, reviewer, journal, your is skilled. Response to modify later, do additional experiment or data analysis if needed. Most of the time they say, do this experiment again. I want this data, I want that data. Reviewer, you have to give that. Make appropriate revisions, point by point response. Sometimes you have, I have seen the responses of the people. Okay, it is done, it is done, it is done, it is done. No, not acceptable. You have to mention what has been done, where it has been done, page number, line number. You have to mention. Otherwise, respond to later, intent of suggestions, explain any additional changes, return revision in a timely manner. Don't keep revisions for months and months, weeks and weeks. Otherwise, paper will be rejected. Okay, so this is suggestion for all of you. Okay, it is happening. So it's a major challenge. You keep revisions for 1 week, 2 week, 3 week, 4 week, 5 week. They have given 5 weeks, 4 weeks. Some journals gave 15 days. Day before study I got a decision. They have given only 14 days to revise. Okay. So you have to revise in 14 days. Otherwise, you may lose the opportunity of publishing because there are so many authors in the queue. More on rejection. This is very important. If your paper is rejected, your work is rejected, your idea is rejected, you are also rejected in an interview, okay? And you are unsuccessful, don't take it personally. Look for the good points and build on them. Editor can help resolve conflicting reviews. Don't pretend to fix something you don't have. If sending to a different journal is the previous rejection is relevant, be diplomatic. Be diplomatic. Don't take it personally. Try to improve it. Okay, because if there is no problem, why the journal will reject your paper? Think about it. And you are sitting here in Banaras, editor is sitting in Sao Paulo, Brazil or New York or somewhere else. Why they will reject your paper? Do they know you? They reject based on your methods, presentations, idea. Not feasible, reject. So please, more on rejection. There are more slides on rejection, but I am not showing that. Okay. Why a manuscript get rejected? You just see. Inappropriate, which I told you. Research is confirmatory only, study is incremental only, study is poorly designed, conclusions made are not supported by data, manuscript written was poorly written or organized. This information is present in the reviewer comments. You can read that. Ethical consideration, ethics are very important. Your name and integrity is very important. Authorship, when it is decided who are the authors, who is an author, what are the responsibilities of author, how it is decided, what are the career implications, that is very important. Authorship should be properly designated. What constitutes authorship? Why you want to become, when your name is included in authorship, contribute to conception, design, acquisition of data analysis and interpretation. Then you can be the author. It is not that you are standing and observing and you will get the authorship. Okay, don't try to be that like that. Okay, because sometimes the student they feel, okay, especially the MSc trainee student, they feel, okay, I helped in this experiment, I should be the author. Okay, you can be author, but if you have contributed that, involved in drafting and revising the manuscript. I told you a recent experience. The paper came for revision. the author, the second author, third author got afraid. Okay, I cannot do that with this that. Okay, so these are all real things. Okay, real life examples. Okay, approve the final version. Okay, then an author must be willing to take public responsibility of the content of the manuscript. The first author who is doing all the experiment should take public responsibility. Public responsibility means you don't blame your mentor. Okay, that okay, when something is wrong in the paper, you tell okay, Professor Shukla is there. Okay, you talk to Professor Shukla. Okay, or you talk to Dr. Mandi. Please be careful. It is your public responsibility. You should be able to take. This method was wrong. I did this wrong. I should be blamed for it. When you get a celebration for your paper, you should also be responsible for it. Please remember. And you should be billing. Otherwise, don't become the first author. Okay. Author responsibility, first, second, whatever author you are. Okay. Defend the intellectual content, including the results and conclusion. Be willing to concede publicly for any errors. You should accept this error happened. Okay, there is no problem. In case of fraud, be willing to state publicly its nature and extent and how it occurred. This is on a bad way, but normally this is a very ethical thing. Please remember this. Authorship listing, decide very early. Don't decide authorship in the last. Okay, you decide early that this will be our team 1, 2, 3. Don't decide the work in the beginning. You can assign the work between, but decide your team that I will take help from 1, 2, 3, 4, 5, whatever you want. Decide it so that your team is there. Okay, principal investigator, last author, lead experimenter listed first, those who are doing the first experiment. The mentor will be the corresponding author. Okay, he or she will be always corresponding author, mentor or supervisor. Dual first authorship is also possible. If two persons have done work equally, they can be also the first author. 2 or 3 persons, no problem. Authorship problems, I am not telling you here, but it happens. Okay, many times, see, the names are deleted. Okay, and you will feel, so these things are to be written and taken care of that. Okay, sometimes you have not contributed, you contributed a paragraph, for example. Okay, and you think you should be author. So you should be briefed by your supervisor, mentor, your colleague that okay, you have contributed this much, so we are acknowledging it. You cannot, I told you the authorship significance. Okay, why ethics important? It is there. Maintain public trust, ensure proper use of funding, protect research, ensure accuracy and reliability, aid collaborations, promote scientific integrity, proper credit of their work. It's very important. Bulletproof your work. This is a very good slide. Okay, your work should be bulletproof. Nobody can steal, nobody does that, and your reading should be very much improved. Okay, so if you have this type of issues, it will, the bulletproofness will be going on. This is a plaque. Okay, so if you don't bulletproof your work and it is like this, it is showing 32% plaque. Okay, some data, some analysis, 32% plaque. It is not acceptable. Normally 8% to 10% in research paper, review paper also 10 to 12 maximum. And I am seeing sometimes you copy a report or something, and don't be smart that you will take AI help or something tools and you will create a section, you will write some section, easily identified. There are softwares, they will identify the boat also, what boat you have used. Okay, and I have shown examples to my students. Okay, that this boat has been used by this researcher, this name is HK123, and you can think that how he knows it or the editor. So please be very careful and don't plagiarize, don't copy paste. Common forms of academic plagiarism: mosaic, paraphrasing, self-plagiarism. These are the 3 common things what most people do, and they do unknowingly also. Okay, it is not that you want to do that, unknowingly also. So include data from various resources and mix them. Paraphrasing, okay, and paraphrasing is also very common sometimes. Then self-plagiarism, I told you in the beginning. Okay, that is also to be avoided. 7 ethical principles to be kept in mind: follow guidelines, protect your subject, don't cheat, don't steal, state true authorship, submit ghost and gift authors. Okay, this is also very common, ghost gift authors. Then submit ethically, avoid conflict of interest. If you really want anybody to include in your authorship, why you don't assign the work? You assign the work, ask that particular person to give the work. Maybe it's a small work or a big work. It signifies your research, improves your research, it is good. So, means asking anything for work and keeping the name is called ghost author. If you have not contributed anything. So, try to avoid that. Avoid conflict of interest. Okay. Never hack the peer review. That is also there. Exercise. This is exercise. Do you come across conflict of interest among group members resulting from authorship issue? It is happening. Exercise करने की ज़रूरत नहीं है. Types of papers. Okay. All these are called as publications. Okay. But mostly review articles, editorial, book review, letter to editor, book chapters, lay writing, along with the research paper, original research paper. So if you are starting with all these editorials, you will not get, because at a senior time you get it. Review also at a senior time you will get. Review article is an original article. I am not telling detail here about that, and because it may take much more time. Book types. Okay, this is a PLOS writing center. I don't know, it is openly available. You can Google it. PLOS resources writing center. I can share the link in the group, WhatsApp group. You read it, okay? They will tell you many things which you have, but not other things which I have covered, okay? They will tell only the polished way, okay? And these are few reviews which we have published. You can see the title also, the author list also, collaborations also, okay? And if you see bioprospecting of functional cellulases from metagenome for second generation biofuel production, a review. This is in collaboration with these people, and you see the timeline. Received 10th October 2016, revised after April 26 April 17, I got decision. How many months? October, November, December, January, February, March and April. 6 months, 7 months. After 7 months, first decision. Okay, patience. Then after that, 1 month for acceptance. So whole cycle you see, and published June. So this is one thing. And the collaboration you see, he is a professor of enzyme technology in Greece, Athens, Agricultural University of Athens. Because he was having very good expertise, so we collaborated in this review. He contributed, and then we made a very excellent review. This is another review. You can see here. Okay. I think timeline is not here, but I can show you. No, it is not here. Okay. Enhancing production of microalgal biofilaments through metabolic and genetic engineering. Okay. This is another review. These are all written by our group. Okay. So this one, gene editing, I shared this one. No, I didn't share it. Whatever I shared, it is not here today because I already shared, so no repetition. Gene editing and genetic engineering. Here also timeline I missed to copy, but you can see most of the time 8 months, 10 months, 6 months. Sometime it is fast also. Then this is the book. Book takes 1 hour, 1 year or 2 years. Okay, it may take 2 years also. The whole book is written. It is available in the library. You can see this journal, this paper, research paper. You can see approved de-inking, bio-bleaching. You can see here. Okay, timeline is almost three months. Here also you see. Okay, received 13 June, revised 1 September, accepted 13 September. I think Mandir paper. Okay, he is the first author. He has done most of the work and he challenges. Okay, anything if comes, he will take the responsibility. So this is the thing which you see the publication cycle. 13 June, September, June, July, August, September, 4 months. Okay. Going further, this one. This is a journal which takes so much time these days. Okay. I have no idea, but maybe the timeline is there. I think 8 months. Would do 8 months it took. Okay. This journal is taking 6 to 8 months. Okay. Impact factor is very high, 16 something. Okay. Journal of Hazardous Material, also very good impact journal. Okay, the timeline is not here. Why, I don't know. Maybe I cut it. Okay. Then this is another one. Okay. And then this one. Okay. Current opinion. Then here also, then you see this I showed you yesterday. This one also there. Okay. Here the reviewer name will also be there. Some journals they write reviewer name also. Okay. So you can get that. Then this one is a book chapter. I am showing a book chapter also. Book chapter will look like this. Okay. It will be a book chapter. Okay. So book chapter, review article, research paper I showed all you. Editorial and forum article also I am showing you next. This is a cell press forum article. Forum. You see the abstract? ये abstract है. कितना छोटा abstract.